There is a fruit that crosses the blood-brain barrier. Not the fruit itself, but the compounds inside it. Specific molecules that enter the bloodstream after digestion, travel to the brain, pass through the protective barrier that blocks most substances from entering, and directly affect the biology of the neurons on the other side. This is not a metaphor. It is not marketing language. It is a documented pharmacokinetic property of a specific class of compounds, anthocyanins, that happen to be concentrated in high amounts in one of the most ordinary and widely available foods in the grocery store. The fruit is blueberries. Most people who eat blueberries think of them as healthy in the vague, general sense that fruit is healthy. Vitamins, antioxidants, something good for you. The kind of health claim that applies to most colored produce without particularly differentiating one from another. But blueberries are not generically healthy in the same way that most fruit is healthy. They contain compounds with specific neurological properties that distinguish them from the majority of plant foods. Properties that have been documented in controlled human trials, that have been observed in brain imaging studies, and that translate into measurable improvements in memory, processing speed, and cognitive function in people who consume them regularly. This is what those compounds are, what they do in the brain, what the research shows, and why blueberries specifically occupy a different category from other fruits when it comes to brain health. The blood-brain barrier is one of the most tightly controlled interfaces in human biology. The brain is the most metabolically active and the most vulnerable organ in the body. It requires a constant supply of glucose and oxygen to function, and it is exquisitely sensitive to chemical disruption. Tiny changes in ion concentrations, pH, or the presence of foreign compounds can dramatically alter neural function. The blood-brain barrier exists to maintain the precise chemical environment that neurons require. The barrier is formed by specialized endothelial cells that line the capillaries supplying the brain. Unlike the endothelial cells and capillaries elsewhere in the body, which have gaps that allow molecules to pass relatively freely between the blood and surrounding tissue, the brain endothelial cells are connected by extremely tight junctions that allow almost nothing to pass without active transport. Lipid-soluble small molecules can cross through the lipid membrane of the endothelial cells. Glucose and certain amino acids are actively transported. Almost everything else, including most drugs, most toxins, and most beneficial compounds, is excluded. This selectivity is what makes brain drug development so difficult and so expensive. A compound can be highly effective at modifying neuronal biology in a petri dish and completely ineffective as a treatment in a living person, simply because it cannot reach the brain. The majority of potentially therapeutic compounds fail at this barrier. Anthocyanins are unusual. They are water-soluble plant pigments, the molecules responsible for the blue, purple, and red colors of blueberries, blackberries, red cabbage, and a range of other plants. Most water-soluble compounds cannot cross the blood-brain barrier because the tight junctions block aqueous movement between blood and brain tissue. But studies using both animal models and human subjects have detected anthocyanins and their metabolites in brain tissue after oral consumption. The specific mechanisms enabling this transport are still being investigated. Partial lipid solubility, active transport by specific membrane transporters, and passive diffusion through temporarily disrupted tight junctions during and after digestion are all proposed mechanisms. Neuroinflammation is the activation of the brain's immune cells, microglia, in response to cellular damage, pathogen invasion, or molecular debris. Acute neuroinflammation is protective and necessary. Chronic neuroinflammation, persistent microglial activation without a resolved underlying cause, is destructive. It is associated with the progressive neuronal damage in Alzheimer's disease, Parkinson's disease, and other neurodegenerative conditions, and with the more subtle cognitive decline that accompanies normal aging. Anthocyanins have documented anti-inflammatory effects on microglial activation. In both cell culture and animal studies, anthocyanin exposure reduces the production of pro-inflammatory cytokines, the molecular signals that sustain chronic neuroinflammation, and shifts microglial activity toward more protective, less destructive patterns. Oxidative stress, the accumulation of reactive oxygen species that damage cellular components including DNA, proteins, and lipid membranes, is a major driver of neuronal aging and degeneration. Neurons are particularly vulnerable to oxidative damage because of their high metabolic rate, their limited regenerative capacity, and their dependence on the integrity of complex molecular machinery accumulated over a lifetime. Anthocyanins are potent antioxidants that directly neutralize reactive oxygen species, but their antioxidant effect in the brain may be less important than their signaling effects. Anthocyanins activate cellular pathways, including the NRF2 pathway, that upregulate the cell's own antioxidant defense systems. Rather than simply neutralizing free radicals as a one-to-one chemical reaction, anthocyanins trigger sustained increases in the cell's capacity to manage oxidative stress through its own enzymatic machinery. Cerebral blood flow, the delivery of oxygenated blood to active brain regions, is a direct determinant of cognitive performance. The hippocampus, the prefrontal cortex, and other regions involved in memory formation, executive function, and cognitive processing all require adequate blood flow to maintain their activity. Reduced cerebral blood flow is one of the earliest measurable changes in conditions associated with cognitive decline. Anthocyanins increase the bioavailability and activity of nitric oxide, a signaling molecule that relaxes blood vessel walls and increases blood flow. In the brain, this translates to measurably increased cerebral blood flow to specific regions, including the hippocampus, following blueberry consumption. The hippocampus is the brain structure most directly relevant to the cognitive effects of blueberries. The hippocampus is the primary site of neurogenesis in the adult brain, the production of new neurons from neural stem cells. While most neurons in the adult brain are formed before birth and last a lifetime, hippocampal neurogenesis continues throughout adult life at a rate that is meaningfully modulated by lifestyle factors, including exercise, stress, sleep, and diet. New hippocampal neurons are critical for pattern separation, the ability to distinguish between similar memories and similar sensory inputs. Declining hippocampal neurogenesis, which occurs with normal aging and is accelerated by chronic stress and poor metabolic health, is associated with increasing difficulty distinguishing between similar memories, the tip of the tongue phenomenon, the difficulty remembering which conversation mentioned which detail, the conflation of similar past experiences. Anthocyanins in animal models increase the production of brain-derived neurotrophic factor, BDNF, in the hippocampus. BDNF is the primary molecular signal for hippocampal neurogenesis and neuronal survival. It is also the molecule that physical exercise increases in the brain, the mechanism behind the well-documented cognitive benefits of aerobic exercise. Blueberry-derived anthocyanins appear to activate many of the same molecular pathways in the hippocampus that exercise activates, increasing BDNF, supporting neurogenesis, increasing cerebral blood flow, and reducing neuroinflammation. The magnitude of the effect from dietary intervention is smaller than from vigorous exercise, but the two effects are additive. Blueberries and exercise together produce larger hippocampal benefits than either alone. The human clinical research on blueberries and cognitive function is more extensive than for almost any other single food, and its findings are consistent enough to merit specific examination. A landmark study published in the European Journal of Nutrition enrolled adults between the ages of 60 and 75 and provided them with either freeze-dried blueberry powder equivalent to a cup of fresh blueberries per day or a matched placebo for 12 weeks. At the end of the 12-week period, the blueberry group showed significantly better performance on measures of episodic memory, the ability to recall specific past events, and significantly faster cognitive processing speed compared to the placebo group. Brain imaging data from the blueberry group showed increased activity in brain regions associated with memory consolidation, suggesting that the cognitive improvements were accompanied by measurable changes in neural activity patterns, not just subjective improvement. A subsequent study from King's College London enrolled healthy adults between the ages of 40 and 55, a younger population than most blueberry research had previously examined, and found that daily blueberry consumption improved working memory and attention measures over a 12-week period. The finding that benefits appeared in middle-aged adults rather than only in older populations suggests that the neurological effects of anthocyanins are not limited to compensating for age-related decline, but may be relevant to cognitive performance in healthier, younger populations as well. Research at the University of Cincinnati found improvements in memory function in older adults with mild cognitive impairment, a condition intermediate between normal aging and dementia, following daily wild blueberry supplementation. The mild cognitive impairment population is particularly significant because it represents the window during which interventions are most likely to affect long-term trajectory before substantial neuronal damage has accumulated. The consistency of findings across different populations, different methodologies, and different research groups strengthens the interpretation that the effects are genuine and not artifacts of specific study designs. Fresh versus frozen versus freeze-dried blueberries is a practical question with a clear, research-based answer. Anthocyanin content in fresh blueberries is highest at peak ripeness and declines during storage. Fresh blueberries stored in a refrigerator for more than a week show measurable anthocyanin loss. The timing from harvest to consumption matters. Fresh blueberries consumed at peak ripeness have higher anthocyanin content than fresh blueberries that have been stored for extended periods in the retail and transport chain. Frozen blueberries surprisingly often have higher anthocyanin content than fresh blueberries purchased from a grocery store. Freezing is typically done immediately after harvest at peak ripeness, before significant anthocyanin degradation occurs. The freeze-thaw process itself causes some cell wall disruption that may actually increase the bioavailability of anthocyanins by making them more accessible during digestion. Freeze-dried blueberries, the format used in many of the clinical studies, concentrate the anthocyanin content relative to fresh weight, since most of the water is removed. 1 tablespoon of freeze-dried blueberry powder contains roughly the anthocyanin equivalent of a quarter cup of fresh blueberries. For people who want to consume consistent, measurable doses of blueberry anthocyanins for cognitive purposes, freeze-dried powder mixed into yogurt or smoothies provides more predictable dosing than fresh fruit of variable ripeness. Cooked blueberries in muffins, pancakes, or other heated preparations show significant anthocyanin degradation. Heat breaks down the anthocyanin molecules and substantially reduces their bioavailability. Blueberries consumed for their neurological benefits should be eaten fresh, frozen then thawed, or freeze-dried rather than cooked. Wild blueberries, the smaller, darker variety native to North America, rather than the cultivated high-bush blueberry common in grocery stores, have significantly higher anthocyanin content per gram than cultivated varieties. The smaller berry has more skin relative to its total volume, and the skin is where anthocyanins are concentrated. If wild blueberries are available in frozen form, as they commonly are, they are the higher potency option for anyone specifically seeking cognitive benefits. The effective dose in the clinical studies that showed cognitive improvements was consistently in the range of 1 to 2 cups of fresh blueberries per day, or the equivalent in freeze-dried powder. This is a larger amount than most people consume as part of their regular diet. The average American consumes blueberries primarily as an occasional addition to cereal or yogurt rather than as a daily staple at therapeutic dose. Whether smaller amounts consumed consistently over longer periods produce comparable benefits is not yet established in the research. The dose-response relationship for anthocyanin cognitive effects has not been fully characterized. What is established is that 1 cup per day for 12 weeks produces measurable, statistically significant cognitive improvements in controlled trials. Doses substantially below this may produce benefits, but the evidence base for smaller amounts is less robust. The timing of consumption relative to cognitive tasks may also matter. Research on flavonoid-rich foods, including blueberries, suggests that acute consumption, eating blueberries immediately before a cognitively demanding task, can produce measurable improvements in blood flow to the prefrontal cortex and corresponding improvements in performance within 2 to 3 hours. Both the acute effect and the cumulative 12-week effect are documented, suggesting that blueberries may offer both immediate and long-term cognitive benefits through overlapping but distinct mechanisms. Here is the synthesis. Blueberries contain anthocyanins, plant pigments that are unusual among dietary compounds in their ability to cross the blood-brain barrier and directly affect neuronal biology. In the brain, they reduce neuroinflammation, support the cell's own antioxidant defenses, increase cerebral blood flow to the hippocampus and other cognitively important regions, and activate the same BDNF pathway that exercise activates in support of hippocampal neurogenesis. Controlled human trials demonstrate measurable improvements in episodic memory, processing speed, and working memory in both older adults and middle-aged populations after 12 weeks of daily consumption at approximately 1 cup per day. If this changed how you think about the blueberries in your refrigerator, hit the like button. Share it with someone who thinks all fruit is roughly equally healthy.